All right, I tend to have a quiet speaking voice, so if you can't hear me, please just wave at me while we're going. All right. So just by way of introduction, I've been working with sequencing data since about 2008, 2009. Most recently, mostly focusing on RNA-seq, but I've worked in 2 different core facilities, one for a commercial company and one for a nonprofit medical research organization before I came here. So I might be making a few comments about back in the day, we used to do it this way. It just means back about 6 years ago, not really back that far. All right. So this may be a lot of review for some of you, and hopefully there's some things that are new to you. But please just, if you have comments, feel free to stop me and ask questions or make comments as we go along. I just wanted to walk through the overall process that we go through from your sample and to the point where you get quantitative values for genes, and talk a little bit about the different steps and their importance for the data that we eventually get out. So, so I'll walk through this for a minute. First, we're going to start with your sample and how that gets, and I'm going to describe basically the Illumina technology because that's the one I'm most familiar with. So how that gets from your sample to an image, from an image to sequence, and from sequence to gene quantitation. Okay. So if we were doing RNA-seq, obviously we wouldn't be starting with genomic DNA. We would be starting with RNA. But keep in mind that's just kind of a placeholder for whatever you start with for your sample. So the first step is just getting from your sample to an image. How do we do that? So first, you use some protocol that's going to isolate your RNA or DNA, and then you need to fragment it into small fragments. This seems like a small step that we gloss over, but in RNA-seq you can fragment a few different ways, and how that affects your data in the end is often that will lead to a different type of coverage across your transcript. So you might be more biased in your coverage toward the 3' end, or more even coverage, which has implications for what you want to do with the data downstream. So there's a few different options for fragmentation. Then we add linkers. Just by show of hands, who's familiar with this the Illumina chemistry here that does this procedure? Is anyone familiar with this? Several people? Some people are? Yes? No? Okay. Sorry, I've got screens here and screens here. I'll stand here. All right, so let me see if I can use this as a pointer. There we go. All right, so we have the adapters here on the fragments. And then when you put the fragments on the flow cell, the little adapters here on the end bind to the flow cell here. And then they go through a process called cluster generation, where a single fragment is amplified by PCR, and it's showing that it's bound on both sides to the flow cell by those little adapters that are complementary to the flow cell. So it goes through a process of cluster generation where we get multiple copies of the same fragment here in one spot. And this is going to make it easier to detect for the detection software. So you took one fragment and you made it into many similar fragments right here. And then it does the sequencing procedure where it goes through and sequences the clusters, and as it adds a base, there's an imaging process where each base is going to release a color, and then it's going to successively take images at each step. So at each step when you're adding a base, you're going to get an image and then another image. And that's the raw data that we get from the sequencing, is just these images. And the images are just showing you the color that's coming from each cluster. So the idea is that ideally, if it works perfectly, at the position where a single cluster is, you should very cleanly get a single color per base, because it should represent one base. So if it worked very perfectly, you would just be able to look at the images and see what base was added at that step. all right, so what I want to say about this part also, so the input obviously is your sample. The output are these big TIFF files. And it used to be when we first started using the sequencing machines, and people started to generate these massive files and then all the downstream files, there's a lot of hand-wringing amongst people that worked with sequence data about how to store all the data because it was very overwhelming. But the technology has changed a lot in that time, and so now the sequencing instruments process those images on the fly, and we don't even have to worry about storing them. So one thing that has been interesting for me to watch is how Illumina has optimized their chemistry and their software so that their cluster generation steps can pack in clusters more tightly so that it allows you to get more fragments on a chip and separate them more cleanly so that you can get more data out of a single run on a flow cell. I think that's all I wanted to say about that. Any questions about that step? Feel free to just wave at me. This is set up so that this monitor is, I can't find my mouse. There it is. Okay, I'm just going to leave my mouse over here. Okay, so now we have these TIFF images that are the input for the next step. And I apologize that those two figures there are not really legible, but what I want to illustrate is that you go from the step of an image, and now we need to turn it into sequence. So the next step is software that's going to take your position and look at the image, look at each position, and convert the values there into intensities for each colors that fluoresce. And it's showing here the 4 colors for the 4 bases. So you're going to get a numeric value at each cluster location for each base, and then that is converted into sequence. So then what you get on the far right are your FASTQ files. Has anyone ever looked at a FASTQ file? A few? So one core I was working with, they were really excited to get this was back in the days of the Illumina GA, and they were so excited to get it and to work with the data. And they would say, like, let me see the data. And I said, well, it's really boring to look at, but I'll show it to you. And they're like, I really want to see it. And so I showed them, and they went, oh. Because it's basically just a big text file, and the header line of the read sequence, the header line again, and then the quality of each base call at each position. And that's the result of what you get out is in a FASTQ. So the FASTQ includes both your sequence and the quality of the call at each position. And this step going from image to sequence is usually done by Illumina's commercial software. There has been some method development on alternative ways to call bases from the images, but usually this is done with commercial software.
How much has that changed over the years? Like, I mean, if you look in the last 3 or 4 years, is that process changing, or is Illumina pretty much still one methodology?
So their software I think their software for this part has become much more stable. In the last couple years, I haven't been paying as close attention, but it used to be that there were major releases that were major bug fixes every quarter or so, and lately it's very stabilized. They don't have major releases anymore.
Just to follow up to that question, how would that would seem to introduce potential batch effects between a study that was run 3 years ago and a study that was run today?
Right. So that can be. So if you have old data, you could potentially, depending on what form you have it in, you could potentially reprocess it with new software. It depends on if you only have the FASTQ, then you have to just start with a FASTQ, and then you would have to model the batch effect in the downstream analysis. But if you have some of the earlier files, you could reprocess them with the latest software, so that you could remove that batch effect. So there's a few options, but yes, it could be different. And if you had data from a GA2 and a HiSeq and a MySeq or something like that, you would probably want to put in a batch for those, or I guess it would be a technology covariate for that data set. Other questions? people used to talk about, one thing we used to talk about also is saving all of the raw data. As scientists, we always love our raw data. And we had a protracted battle with legal once about being able to dispose of the intensity files and just keep the sequence files at the company I worked for. But another evolution I've watched is that now often people say, it's cheaper for me to just resequence my sample if I need to, rather than to store all the data that we used to have. And so that's been, as long as you don't have super precious samples, more people are going to that. We would just rather resequence than have to worry about storing these, because it's a massive operation to store all of the data. Okay, so then the next step, which is probably one you hear about a lot more, is going from sequence to gene quantitation. Now, you can quantitate at different levels than gene. I've just mentioned gene here to be simple. And I realized sometime late last night or this morning that I don't actually have a slide in here that describes the process of alignment. So we can talk about that a little bit. But what you start with is your FASTQ sequence reads. And what you want to get is you want to know how many of those reads came from which genes or transcripts. So the way this is typically done is you do an alignment. I can't mess with the mouse, so I'm just going to point with my hands. So you do an alignment of, so these are meant to represent the reads. I imagine everybody's looked in a genome browser. Oh, technology. Yay, thank you. All right. Has everyone looked at a genome browser? You've seen this kind of visualization of reads somewhere, so you're kind of familiar with it. So the idea is that you take all these reads and you want to know where they came from, so you match them up with where they go on the genome. So then you have this location, and this is just showing you a whole bunch of reads came from here, 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 here. And this coverage is quantitating how many reads were at those locations. So then we just know you have a genome, and you have a bunch of reads that came from different places. And then what you do is put the gene models on top of that, and then you just say, okay, in this region, let's say this is one gene. In this region, there are this many reads. And we count them up, and that's how many there are. And in this region, there are that many reads. And we count them up, and that's how many there are. And in this region, there are that many reads. So that's the really simplistic way that you can get from sequences to quantitation. Now, when I say simplistic, that is the most simplistic algorithm you can use. There are much more complicated ones for inferring the quantitation of transcripts that I won't go into completely now. But that's the basic idea. You want to match the reads up with where they came from on the genome or transcriptome, and then quantitate that to gene counts. So what you get from this, and some of you may have seen this, you'll get usually in a text file. It's often just a matrix that has genes in the rows and samples in the columns, and it has a value for each gene. And then you also may have heard of BAM files. People familiar with BAM files have heard of them? Some people? No? Yes, okay. So a BAM file it contains the reads and the information about their alignment. And there are a lot of software tools that work with the BAM format because it's a very common format for holding sequence alignment data. So there's a lot of tools that are specialized to get data out of the BAM format. All right. So the number of tools that you can use to do an alignment keeps growing. That's what, as bioinformaticians, we like to do, is invent new or slightly different variations on a way of doing something that's a little bit faster and a little bit better. So there's a lot of alignment tools. And these steps that I described previously, there's also a lot of variation on those steps. So a question that we often ask is, what's the optimal protocol for doing RNA-seq? Yes, go ahead.
Can you say something about what the initial QC you want to do when you first get sequencing data back would be? Like, what would you want to look at specifically? Q30 scores or duplication rates, or what would your checklist be?
Right. So if it's RNA-seq, I usually look at the alignment rate. I look at the fraction of genes that are detected across all the samples. I look at the fraction of biotypes, how the reads are allocated to the different biotypes, like how many are protein coding, how many are pseudogenes. Those are some of the basic things. But then there's a lot more depending on, I guess, what the data set is like. Why don't I go through the next part, and then can we come back to that a little later, and perhaps we can make a list of things that we've all done for QC. There's quite a range of things you can do. You can check for contaminants, and some of it's a little bit specific to your protocol and what you want to get out of it. And I could also afterwards send you kind of the list of what I typically do for QC if you're interested in that. So, all right, so we'll come back to that in just a minute. Make a mental note. I wanted to talk for a minute about the optimal protocol for RNA-seq. I'm sure if you're all from different places and you had samples, everybody's going to have their samples prepared and processed differently. And it depends on who your collaborators are, or what methods you want to choose, or what tools you have available to you. So I wanted to talk for a minute about how we make those choices. I'm going to use some data from a few papers. They've been out for a few years, but I think that they're good. They're good comparisons of the different options that are available. So if you're familiar with these papers, please jump right in with your comments as we go through this part. So the first one is about RNA-seq alignment. So I'm sure that some of you have anybody used, has anybody used an RNA-seq aligner or worked closely with someone who has? Yes? Can we throw out some names of any aligners we've used or familiar with?
How many has used one? Come on, hands up. Anyone? Okay, no one has used it.
Okay. But you've worked with someone who has used it, perhaps? Yeah? Right. Top Hat. Anybody else? Star? Good. So there's a lot of choices. And which one to choose is a good question. So what they wanted to do in this study is basically just take the same input data set of sequence reads, align it with a bunch of different aligners, and compare the results. And the idea is to give you some idea of if you're going to choose an alignment method, why choose which one or the other? You know, how do they compare to each other? So this was done with paired end data, 76 base pair reads, which is quite a good length for doing this kind of work. And then they had these 4 data sets. So, whoops, I think that's not 562. I think it's something else. I may have written that wrong. 625, I can't remember. Anyway, cell line data from whole cell, cytoplasmic, and nuclear isolates in duplicate. Then they had a mouse brain sample and 2 simulated human data sets, and these are the depths. And the main difference between these 2 simulated data sets is they simulated them such that in this case, 20% of the expression came from novel isoforms, and in this case, 35%. So these are things that wouldn't be represented in the gene models. Let's see. And then all of those algorithms were, all the alignment algorithms were run by the developers. And there were 9 teams that participated. They didn't know what the true alignments were, and they didn't have access to other teams' submissions. So they basically made their best effort with their method and submitted their results. Okay, so I'm just going to show a couple results. This is a really interesting paper. If you'd like to look into it more, there's several more figures to go through. But I just want to go through a couple. So alignment yield. So basically, here we're just looking at how many of the reads could they align. So down on the bottom here, it's fraction of mapped fragments. And uniquely mapped and multi-mapped are 2 basic concepts in alignment. Uniquely mapped means you have a sequence read, and the aligner was able to place it or find its best hit to 1 unique location in the genome or transcriptome. It had a single best hit. Multi-mapped means that its best hit was equally good to multiple places. So possibly it contains some repetitive sequence, or maybe it's very short, and so it matches several locations in the genome or transcriptome equally. So here, what we're looking for is ideally a nice large bar of this blue, saying that it's uniquely mapped. So these are the alignment tools. Oh yeah, 562, that is right. Okay. And how they performed on the different data sets. So right away, you can notice a few differences. There are a few groups of software that all get about the same level of alignment, of unique alignment, up here, here. You know, there's quite a few that are getting high, and then there's some that are down, you know, much lower. And you can look over here, those trends are pretty consistent. So the basic thing I take from this is that there are performance differences in these tools that matter because you're getting a larger fraction of your reads aligned. And that means you're going to have more data that goes into calculating your gene quantitation. So I actually use this quite a bit when I'm trying to pick an aligner myself. You know, we want to get a lot of reads aligned, but then we also want them to be accurate, and we want the software to be fast.
Quick question.
Yeah.
And maybe you'll go over this. I don't want to. If you are, just let me know. Is this specifically genome map reads, or is this genome and transcriptome, and is this junction map included?
Let me see. I'm trying to remember if they did a genome or transcriptome alignment. That might vary based on the tool. I think so, because they work differently.
Yeah, and I only say that because I've done very limited testing.
Right.
To make sure STAR was working as well as top half before I moved to it, because it is way faster.
Right, exactly.
Has larger memory requirements. But the thing I was thinking about, I thought like I think it's 20% less reads with junctions for STAR. So I haven't done as a great assessment of that. But
Yeah.
that's what I've seen in a few tests that I've done.
I didn't put in the figures here about the junction reads. They do have some figures about that, but it's not shown here. So this isn't particular to junction reads. This is just total fragments. Yeah, they do split out differently about how they handle junctions. And so that's an excellent point, too. If you're really concerned about handling the junctions, by which we mean a single read from a single transcript may cross the junction between two exons. And so that makes it tricky to align and figure out where it came from, because you need to align across that junction. So if you're very concerned about getting those junctions correct, you might care more about which reads cover junctions more effectively as opposed to which ones have a total yield that's higher. Okay, so accuracy. They can only calculate accuracy for the simulated data sets because those are the only ones where they know the true alignment locations. And so here, dark blue is perfectly mapped, light blue is part correctly mapped, and then the red is mapped, but no base is correct. So obviously, I think they colored it red for a reason. Large red bars mean this method has issues, and you might not want to use it. Or you might want to be very concerned about that, because it's aligning a lot of reads, but none of them are to the correct, or none of the bases are correctly aligned to the correct location. So, but again, we're seeing, you know, a fairly consistent amount of perfectly mapped reads here among most of them, and then there are some that are much slower. So again, if you're picking a method to use, as far as accuracy and yield, you know, there's a group of tools that are getting you know, pretty good accuracy, pretty good yield, and then the next question you probably want to ask is how fast are they, so we can compute the data. All right, I think I'm going to skip the next one in the interest of time. Well, I'll just mention it's super fast. This is just a profile of, I know you can't read it very well, but these are the alignments, and then this is a profile across the length of the read about where mismatches occur, insertions, deletions, and introns. And I'm not going to go into detail into this other than just to note that you can see, because the shapes of these are different, that they're handling mismatches, insertions, deletions, and introns differently. And so this is why you're getting different performance from the different methods. So like this one is getting a lot of insertions at the ends of the reads. This is a little more consistent. Anyway, I'll just mention that really briefly. OK. So I think I said all of these things. Yeah, there are tiers of aligner performance. So not all aligners are created equal. Some of them do perform better than others. The most common algorithm, though, the one that you might hear of or see most often, is not necessarily the best one. And then the other thing I didn't point out is that use of the gene models. So if you're working with human data, presumably, or any other species, using the known gene model annotations really improves the performance of the algorithms. Yeah.
I don't know if you're going to go into this.
Yeah.
But how do you evaluate that in terms of when you're analyzing data or data set for the first time? Which workflows you're going to use, and do you use one or do you use multiple?
What aligner am I going to use to work with my data? Right. So in most cases, I don't choose it per data set. In most cases, I've been and I sort of have a common workflow that I'm using that's based around a certain algorithm. And so in most cases, I'm looking for one that's general purpose. And so in that case, I look for a combination of something that's most accurate and then most fast, usually is the compromise that I'm trying to pick. If I had a special case, like for example, if I want, GSNAP is one that will align with a SNP database in mind, right? So if I was really concerned about data that had a lot of SNPs in it, and I wanted to be able to align those reads more accurately, I might go with that one for that reason. But I tend to not switch aligner to aligner because for me it's important to have a record of the performance of the aligner over time, and so that I can understand better where it's if it's not working very well and I'm not getting very good results from it. If I haven't used it very much and it gives me erroneous results, I might not be able to find those as well. Sorry, that was a long answer to a short question. Yes?
You may cover later, but do you have any preference for the study of the variants, alternative splicing, or any variant alleles to study instead of looking at the quantifications? Because alternative splicing for other alleles that may be different than looking at the quantification.
Oh, so do we have tools that are for analyzing the alternative splicing instead of quantitating? Sure. So usually the way I would handle that is I would first quantitate the transcripts. Are you talking about novel transcripts or just alternative isoforms? Right. So for that type of analysis, there are two kind of approaches that I take. One is that I'll quantitate the data at the exon level, and then I will use a method that looks for differential usage of exons, which is kind of one way to get at different use of isoforms. Alternatively, I'll quantitate the data at the transcript level and then use, there's a series of tools that will look at differential usage of isoforms. But I, in both cases, do the quantitation first, and then the downstream statistical tool will analyze the alternative splicing. Were you looking for some names of tools that do that?
We'll go through a list tomorrow.
Oh, great. OK. OK. So then this is another study that I just wanted to mention. And I thought this one was really interesting because they compared many variables in their study. So they compared the technology platform, the library prep type, RNA degradation, and lab site. And let me just go through this. This is a complicated experimental diagram, and I've even cut off half of it. So it's even more complicated than this. But let me just highlight a few points. So the basic study is there are two samples, A and B, I think this one is the universal human reference, and this one is like a human brain sample. So they're commercial samples that you can purchase, and a lot of people use them just for reference studies. And then these are some synthetic transcripts. And they were mixed to generate samples A, B, C, and D. So sample A is from A with a little bit of the synthetic in it. And sample B is from B with a little bit of the synthetic in it. Then C is a mixture of A and B where there's 3 parts B and 1 part A. No, I'm sorry. I said that backwards. Follow the diagrams, not my words. 3 parts A, 1 part B goes into C. And then 3 parts B, 1 part A goes into D. So they did that, and then they sent these samples out to multiple different labs to run the same samples. So they were sequenced on a PacBio machine, a 454 machine, and 2 types of ion machines, and also on an Illumina machine. So they can compare across all those different platforms. So RNA sequencing has really expanded in the options that are available for sequence now. And then with the Illumina, they used a poly-A enriched protocol. So that's going to select for your protein coding genes. They used a ribodepletion protocol. If you're not familiar with this is one where you're going to prepare your library, and then it's going to deplete out the ribosomal, or remove the ribosomal reads from your sequence. So because presumably we're not interested in the ribosomal reads, and then hopefully what it leaves in is what you're interested in. And then they also tried different ways of degrading the RNA to see what the effect of that was. So very complicated, but they have some interesting results, and I'm just going to highlight a couple of them. Okay. So coverage of your transcript. So what you would ideally like to do is have reads that go across your whole transcript so that you can effectively quantitate it, and you want, and you're not, particularly biased toward one end or the other. So this is really, I thought, an interesting plot showing how that changes across the technologies and with the preps. So the basic data that we're looking at is, here we're looking at coverage. And this is the 5' end of the transcript and the 3' end. And these are just little bins across the transcript. And when it's dark, that's we're getting a lot of coverage on that part, or when it's red. And when it's blue, we're getting very little coverage on that part of the transcript. So a really common protocol to use is Illumina, intact, poly-A, prep. And so if we look at that, would you say the coverage is uniform or biased one way or the other? Biased toward which end? Yeah, 3' biased. Yep, right there. Okay, so let's compare that to the Illumina intact ribodepletion protocol. Thoughts on how they compare? So this one is a little bit more uniform, not quite so biased as this one. PacBio is, if you look over here for a comparison, is quite uniform in coverage across the whole transcript. The LifeTech data here seems like it's more strongly biased, maybe, than the Illumina data. So you don't need to commit all this to memory. But the point I want to make is that if you have reasons to be strongly concerned about uniform coverage across your transcripts, it's something to think about. They do vary in what the result of this is going to be on your coverage. So I've worked with a lot of Illumina data, and this type of bias is pretty normal. I've seen that a lot. Oh, these are different sites. But these sites, as you can see, didn't get quite as much bias. So even from site to site with the same protocol, you can get it a little more even. Anyway, questions about that? Yeah?
I'm sorry. When you say bias, what does that mean?
Oh, sure.
Looking at different genes on top, and then these different colors, I wasn't completely sure.
Yeah, sorry. I'm sorry I didn't explain that so well. So this right here is the length of your transcript from your gene that you're interested in. So this is like the beginning of it and the end of it. And what you're interested in is how many reads you got that mapped to the different places along that transcript. So when you did the sequencing, how many reads did you capture from the beginning of it? How many reads did you capture that came from the middle of it? How many reads did you capture that came from the end of it? And what this is showing you is that in this case, since it's blue, we have very few reads from the beginning of that transcript. So we can't quantitate the beginning very well. And if we wanted to do anything like look at variation in the structure of that transcript at the 5' end, or look at maybe its start site differences, we wouldn't be able to do that very well because there aren't very many sequencing reads from that part. However, we do have good coverage across the middle. So we have a lot of reads in that transcript that came from the middle of it. So it's just when you were preparing your library from your sample, how uniformly did you get the reads? Did you sample reads across your transcript is what you're trying to assess. Yes?
What's the difference in the sequencing chemistry between the different platforms that gives you more or less bias?
So I'm trying to think if I can answer that briefly.
Or like, how does the Pacific Biosciences platform work differently than Illumina?
So I haven't worked with PacBio data for a long time, so if there's someone in the audience that has, feel free to correct me. But the idea with the PacBio is that you have a single molecule, and let's see, the enzyme, I could do this about 4 years ago. Is there anyone that's more familiar with the PacBio? You're working with a single molecule, and you're going, I think, is it fluorescing? The enzyme, I can't do this off the top of my head.
The full transcript, I think, is attached to a pore, and that the polymerase is attached to the pore, and as the RNA, the RNA is moving through it, it has a different, you basically get a different fluorescence.
Is that for the PacBio? I thought that was ion, yeah.
It might be, yeah.
PacBio is something about the enzyme moving along the, yeah.
That's right. The PacBio does have, they have a single pore, and they have a polymerase sitting at the bottom of the pore.
There we go.
Yeah. And then they have fluorescent labeling each of the nucleotides.
Yeah.
And as and basically it's a channel, so you get tunneling effect. So like that fluorescence, as it gets right to the pore, is amplified by the shape of the cavity. So they're detecting single molecule events.
Right, but they don't start with a fragmentation step, whereas with the Illumina, you start with transcripts and you fragment them into smaller pieces. PacBio doesn't do that. They're working with a single molecule, so it's more. It's been a few years since I was listening to all the PacBio talks, so I'm not as up on that. Yes?
And also, I was wondering if you can explain why there's so many more samples compare like Illumina versus Roche versus Lifetime. It seems like, Roche has more sequence that they're reading in the middle, but there's only few that it could read versus Illumina, they're reading a whole bunch of more things. And
Oh, sure. Yeah, so
because then you can compare, is this method the better one because the one method could only read a few very well, but not everything, whereas Illumina can read more and some good, some bad. So like, how can you compare if the numbers not the same?
yeah, so this is part of the experimental design, and here is the site. So this is just the number of times that they replicated that experiment. So with Roche, the reason we have so few columns here compared to Illumina is just because there were fewer sites that did this experiment. So they designed it so that they had one RNA type, one protocol, and then 3 sites each ran the samples. Whereas with Illumina, they designed it so that there were many different sites because they were also testing these different protocols and ways of degrading the RNA. So that's just part of their experimental design, that they had different numbers of sites running different numbers of experiments because they wanted to compare combinations of these things.
So should we just look at impact to, like the impact groups then?
Yeah, I think you can just look at, you can compare, for example, the poly-A Illumina samples to, the Roche poly-A samples, and the poly-A samples over there.
By the single columns?
Yeah.
Oh, I see.
Yeah. Okay. In the interest of time, I'm going to do this one and then skip ahead a little bit. One thing I wanted to mention really briefly, and I have seen this in my data, and it has a particular implication that I think goes unappreciated. So just in the Illumina data, I mentioned that you can do a poly-A enriched or a ribo-depleted prep. Those are different ways of selecting for the RNAs that you want to assess. So when you start with your library, and they either select the transcripts that are poly-A tailed or subtract out the ones that are from the ribosome. And when they did that, this plot here is showing the fraction of mapped reads that came from different parts of the transcript. So reads that came from the intergenic regions, which either are noise or they are novel unannotated transcripts, is fairly consistent between these two protocols. But look at the coverage of exons that's different. That's a really big difference there. So a lot of the time when we're doing RNA-seq, we're interested in quantitating protein coding genes and their exons. So this is the main data that we're going to use to do that. And note that the amount that you get varies a lot between these two protocols. So you're getting almost 50% of your data going to exons in poly-A. So that means almost 50% of your sequence data that aligned is to exons. But with the ribodepleted, you're only getting about less than 25%. And the intron content is much higher here because you're getting some transcripts that are not fully processed. So they include their introns. So that makes a big difference then when you go to quantitate your data, because you first start out with your raw sequence reads, and then some fraction of them align. Usually it's a pretty high fraction, so somewhere in the 90% of them align. So about 90% of them align. And then from that, some fraction of them cover the protein coding regions that you're interested in. If you start with, like if you have a limited budget and you start with not a lot of reads, for example, and then you lose about 10% of them to mapping, and then you lose about another 75% of them because they don't cover the exons that you're interested in, then the actual amount of data or reads that you have to contribute to quantitating those genes is pretty low. And so it's something to keep in mind because people will often talk about, this is my RNA-seq data set, and I sequenced it at 50 million reads. Well, that's good, but if you sequenced it at 50 million reads and you had really high alignment and a large fraction of them went to your protein coding genes, then 50 million is a reasonable number to say that you got pretty good quantitation. But if you had low alignment and then only 25% of your reads went to exonic regions, then your effective depth is much lower than that. So your quantitation, I guess, will be less accurate, is my point there. All right. I think I'm going to skip ahead to the humerus part. I didn't show this data, but what I wanted to mention from that experiment is that when they compared the differentially expressed genes between the two samples A and B, they actually found a large amount of consistency even across platforms, sequencing platforms, in which genes were found to be differentially expressed. All right. So we have a few minutes. Okay, who's seen this? Has anyone seen this video? A few? Yeah, I know you have because I made you watch it. Let me see if I can make it work with the sound. I don't know if this will work. This will be an experiment. I have no idea how to adjust the sound either. What? What do you want me to do? I've plugged it in here. Okay, we'll try it and see how it goes. Oops. How do I skip my... Okay. If this doesn't work, I'll just... Oh, here we go. All right.
I have a study, and I need to know how many patients I need. I think I only need 3 patients.
Okay, what kind of study is it?
I'm doing a lab study. Can I just use 3 patients?
That depends. What are you trying to show? Okay, what are you trying to show?
I'm trying to show that drug A and drug B are better than only drug A.
How are you measuring if it is better?
We are looking at apoptosis. Can we just use 3 patients?
What kind of measure of apoptosis? Do you know how variable the measure is?
We always use 3 patients. That is what we have published before.
3 patients per group or in total?
Yes.
Okay, let's try something different. What type of measure are you using? And is this an in vitro study? An in vivo mouse study?
It is an in vitro study of patient samples, and we are looking at measures of apoptosis after treatment with drug A and drug B versus drug A and control, and drug B alone.
Oh, how many groups are you analyzing altogether then?
My grant is due tomorrow. If I can just use 3 patients per group.
Okay, well, you probably should have come to me earlier for help with this.
All I need you to do is tell me that 3 patients are okay.
So, it's not that easy. Your marker for apoptosis may have a large amount of variability associated with it that could make it difficult to ascertain differences based only on 3 patients per group. It also depends on what kind of outcomes you are looking at. Are you looking at normalized changes in expression levels from baseline? 3 patients may not be sufficient to detect differences in these expression levels between the different treatment groups.
I do not understand.
I'm saying that you may need more than 3 patients per group, but that I need more information to determine that.
The serine protease granzyme B is made in cytotoxic lymphocytes where it is inactive due to the low pH of the storage granules. It is measured immediately upon delivery into target cell cytoplasms where the pH is neutral. This is followed by cleavage of phase 3 by granzyme B in the target cell cytoplasm and the subsequent induction of apoptosis. All proteolytic events can be quantitated at the live single-cell level by flow cytometry and imaged by optical microscopy.
I understand very little of what you just said. Describe more about this.
I just need to know if 3 patients are enough. My grant is due tomorrow.
We will need to meet to better understand this before we can determine the appropriate sample size.
I can put you on my grant for 0.5% FTE. You can design and analyze the data later.
Do you really think that is enough to do the work?
Well, it may be less if it is awarded and the budget is cut.
Please go away.
All right. Okay. Let's see. Now I mute. Do I mute it now? Okay. All right. I show that just to illustrate that there's, there's, I guess, big communication gaps sometimes in the different disciplines that are involved in working with RNA-seq. I think both of them had trouble understanding each other. And this is, I've been in conversations not unlike this. And it's, in my mind, it's just a consequence of a new technology coming into an area, and a lot of different scientists are interested in using it. And it takes some time to understand the technology, its strengths and weaknesses, how to design your experiments for it, and how to get good data out of it. For example, early on with RNA-seq, people even thought we didn't need to do replication, like you didn't need to have biological replicates, and that you could just take your biological replicates and pull them all together and run them just in one sample, and that you would magically have the answer to whatever question it was. And I don't think it's the fault of either scientist that this happens. It's just, it's a new technology. It has different properties, and it takes a while to design your data for it. So I'm probably, I may be beating a dead horse here on this topic, but just some things to think about when you're doing an RNA experiment. Think about, what RNAs am I trying to capture? Am I just trying to quantitate protein coding genes? Do I want to get the long non-coding RNAs? What do I want to get? And that might have an impact on what library prep you're being interested in. What kind of output do I want? Am I just quantitating at the gene level? If so, you don't need to do 100 base pair paired end reads at 80 million reads, per sample. That's a little bit overkill. Are we interested in splice junctions and alternative splicing? We can design a little bit differently for that, or transcript quant. If we want to detect novel transcripts or assemble the transcriptome, then you want to do something that's higher depth. You might want to look at more uniform coverage. And how many samples do I need? I'll just show this really quick. So if you have some budget for your experiment, do you spend it on more samples or more depth? This is a question that comes up every time with a new experiment. So in this case, they had Illumina HiSeq 2000 data. They had 50 base pair single-end reads, and they sequenced them at 30 million reads for, I think it was 7 samples. And then, so they sequenced them at 30 million reads, and then they downsampled that, so they had data sets at 25 million, 20 million, 15, 10, and 5. So these are just sampled from the original data set to make smaller data sets. And then what they did, so they're plotting across the number of reads that they sequenced, what is the number of differentially expressed genes that they found with some FDR cutoff. And the different lines represent how many samples they used in the comparison. So in this one, they just compared 2 samples to each other, 3, 4, 5, 6, 7. So what I want to point out here is that with the 2 samples at 30 million reads, they don't even get to the lowest mark of the number of differentially expressed genes that were found using 7 samples and less than 5 million reads. 5 million reads is really, really low for RNA-seq. Like, nobody sequences at 5 million reads. But this is just to show you that statistically, you're going to be able to get a lot more from your experiment if you have more numbers of samples as opposed to fewer numbers of samples and a higher amount of read depth. And there's a lot more detail on this in the paper, but in the interest of time, I wasn't going to go through that. OK, so just a few points. And then if there are additional questions about QC after, I'm happy to talk about those. So Illumina technology uses sequencing by synthesis and imaging to generate the FASTQ data. So that's the process we went through in the beginning. But now, in addition to Illumina, there's a diverse set of technologies that exist to meet a wide range of sequencing needs. So you really can customize your experimental design for your data. But it does, the time you spend upfront planning, you can tell I've been in this conversation a few times at the end where someone is like, here's my data, work your magic and make it tell me what I want. And I look at it and I'm like, but I can't with this experimental design. So that's why I'm harping on this point. So anyway, careful planning is needed to choose an optimal technology. So the bioinformatics tools are in active development. There's a lot of tools, and they do have different strengths and weaknesses, and some of them are better than others. So when you're working with the data, if you get it from somebody else that's processed it and aligned it, ask them, what are the strengths and weaknesses of the tools that you selected? Why are we using these tools? And I'm sure they'll be happy to tell you. Scientists love to talk about their work. And then, yeah, again, fancy technology cannot make up for poor experimental design. Consider your end goals when designing your study. That was all I had. I know we had a question about QC that I can address later. Are there any other questions? Wow, I talked for a long time. Sorry. There was that part in the middle that was funny, though, right? All right. Well, if you think of questions, I think you have my email. Oh, yeah.
One quick question. So this is a challenging one, and I'm just curious what you do about it. You have a data set that you've gotten from the web or somewhere else. You have a data set which you'd like to compare it to. So you have some hypothesis that condition A, treatment with drug A in your data set,
Mm-hmm.
and treatment with drug B in a previous data set are doing the same thing. How do you compare those data sets? Do you do it directly or indirectly?
So by directly and indirectly, do you mean do I try to process them all together and analyze them together? I guess I would look at how close the technical parameters are. Like if they're both from a similar type of sequencing experiment, I might try to put them together and then have some batch effect for difference of site. If they're completely different technologies and very different preps and things like that, I think I would do a differential expression of the data separately and then do some sort of like a meta comparison afterward, probably.
So I know that that's a question I usually get is how do we compare? Like, so we're presenting a lot of data from this progenitor cell biology consortium, which is going to inherently be technologically captured in a very different way. Or not very different, but different enough than the data that's being generated somewhere else because it's
Right.
generated, it started 4 years ago when it's low depth and single entity. So.
Yeah. So, I mean, in that case, if you analyze the one data set and you get, some results of some gene lists, and you can, compare the overlap of the gene lists and things like that. I'm not a statistician, so I can't talk about the latest and greatest meta-analysis methods, but I probably wouldn't try to treat them exactly equally and analyze them together if they're very different that way. All right. Sure. I how to close all these things.